Life on Earth has been shown to be able to cope with some of the harshest conditions our planet has to offer. Microorganisms, in particular, are renowned for their hardiness and are able to cope with some of the most extreme environments on Earth. Heat, cold, toxicity, you name it, they can handle it. But how exactly do they manage this? Well, extreme conditions can mean many things, and each one requires its own set of adaptations for anything to survive in them. We can start with some bidirectional variables. One of these would be temperature. Many organisms can survive at room temperature, or what we call hot conditions. However, temperatures near freezing or boiling are very difficult for any water-based life to survive in. Another bidirectional variable is pH. While neutral conditions are easy for most organisms to live in, those that are highly acidic or highly alkaline are difficult to survive in. Now, what are some unidirectional variables? Well, we can start with scarcities, or the less there is of something, the harder it is to survive in. One of these is water, or adaptations to extreme aridity. Another is oligotrophy, or the rarity of nutrients an organism can use to grow or power its energy metabolism. Other unidirectional variables require adaptations when too much of them are present. One of these is salinity, or surviving in solutions with extremely high salt concentrations. Another is radiation, and the last is toxicity, and this one can be subdivided as we'll soon see. We'll first start by looking at temperature. Perhaps the most famous environment in which extremophiles inhabit is deep-sea volcanic hydrothermal vents. Bacteria, which live near these vents, power their energy metabolism by oxidizing sulfide, using dissolved oxygen in the water column as the electron acceptor. Some ways they cope with the boiling temperatures is to use basic physical biochemistry. One thing they do is to have a high GC content in their genome. Adenine and thymine nucleotides are held together by two hydrogen bonds, while guanine and cytosine are held together by three hydrogen bonds. This means the greater the GC content of a sequence of DNA, the higher the temperature the two strands will separate at, also known as the annealing temperature. Another strategy that thermophilic prokaryotes employ is to use more saturated fats in their cell membrane. Saturated fats have higher melting temperatures due to the lack of steric hindrance from the kink at the site of the unsaturation. So, a membrane made of saturated fats will remain stable even at high temperatures. While bacteria and archaea are ubiquitous throughout all habitable environments, archaea seem to be more abundant in hydrothermal systems than they are elsewhere. Part of this may come down to the different lipids in their membrane. The ether bonds are more stable than the ester bonds used by bacterial membrane lipids. Also, instead of using lipids without unsaturations, thermophilic archaea use lipids without side chains, which also increase their melting temperature. The last biophysical chemical strategy thermophiles employ is to have a larger hydrophobic region within their enzymes. What causes enzymes, which, like all proteins, are linked chains of amino acids, to have the conformation that they do is that the hydrophobic amino acids are on the inside while the hydrophilic ones are on the outside. Higher temperatures will cause enzymes to unfold or denature, and having a large hydrophobic region, i.e., one that's rejected by water, can allow enzymes to remain stable at higher temperatures. In the world of environmental microbiology, there are both thermophiles and hyperthermophiles. Thermophiles have optimal growth temperatures in excess of 55 degrees Celsius, i.e., above the hottest recorded ambient atmospheric temperatures, while having a maximum growth temperature of around 80 degrees. Meanwhile, hypothermophiles require temperatures in excess of 65 degrees to even grow, and are capable of growth at over 100 degrees. The reason water can sometimes remain liquid at these temperatures is due to high pressures or salt concentrations. In accordance with basic physical chemistry, the hotter the temperature, the greater the rate of a chemical reaction, including metabolic reactions. So thermophiles will usually have higher growth rates than microorganisms which grow in ambient atmospheric temperatures, also known as mesophiles. However, once you get past 75 degrees Celsius, the aforementioned biophysical chemical adaptations in the genome, membrane, and enzymes are no longer sufficient to cope with the high temperatures. As such, other adaptations are needed to cope with the extreme environments. These adaptations either directly require energy that could be used for growth or division, or involve a trade-off where the functionality of a molecular component is decreased to allow it to remain intact at boiling temperatures. This causes hyperthermophiles to have lower growth rates than thermophiles. In the enzymes of hyperthermophiles, not only do they have a large hydrophobic interior, but on the surface, they substitute neutral amino acids which hydrogen bond to each other with charged amino acids which ionically bond to one another. Ionic bonds are much stronger and will persist even at high temperatures. Another feature the enzymes of hyperthermophiles have is a lack of loops over the surface and a lack of internal cavities within the enzyme to increase the packing density. Also, whereas other prokaryotes use hydrogen bonds between enzymes in various structures to keep the enzymes localized within a part of the cell, hyperthermophiles use covalent bonding. Hyperthermophiles also have abnormally high concentrations of chaperone proteins, which help other proteins to fold into their desired shape. They also use these to prevent their DNA strands from annealing. Hyperthermophiles also possess a reverse gyrase enzyme that rewinds sections of DNA that have become annealed. At higher temperatures, many chemicals, including oxidants, become more reactive, and because of this, oxidative stress is a much bigger problem for hyperthermophiles. As such, they have to produce enzymes such as catalase that detoxify reactive oxygen species and need to spend more energy repairing oxidative damage. The hottest temperature any microorganism has been documented growing at was at 121 degrees Celsius, and the organism which did this was called strain 121, an archaea. Apart from deep-sea hydrothermal vents, thermophiles and hyperthermophiles can be found in hot springs, along with artificially heated environments in industrial settings. At the other end of the temperature spectrum, microbes have the opposite problems. Microorganisms which inhabit freezing temperatures, known as psychrophiles, also employ biophysical chemical adaptations to cope with the temperatures. Whereas bacteria in hot conditions have fewer unsaturations, those in cold environments have more. Any organism has to strike a balance between having a membrane that's not too runny nor too rigid, and at colder temperatures, this balance has unsaturated lipids with lower melting temperatures. Some psychrophiles also have lipids with more side chains on them, making them more fluid at low temperatures. A common strategy seen in psychrophiles is to upregulate the genes involved in degrading molecules which make the membranes more rigid, such as cholesterol. Psychrophiles which inhabit glaciers in temperate zones exhibit phenotypic plasticity, being able to produce more saturated lipids at room temperature. Quite often, if you look at glaciers, you can see colorful patches all over them. This is from pigments known as carotenoids in the membranes of psychrophilic microbes. Exactly why psychrophiles produce these isn't entirely clear, but there are a number of hypotheses. For one, they may maintain homeoviscosity in the membrane, counteracting the fluidizing effects of the unsaturated fatty acids. Another function that they can fill is an antioxidant, which protects the molecular machinery from reactive oxygen species. Reactive oxygen species are produced by many photochemical reactions involving UV light and water. The rate at which they're produced in cold habitats isn't really different than in warmer habitats, but the lower temperatures also mean the metabolic reactions of psychrophiles proceed at a lower rate, which makes it harder for them to repair oxidative damage faster than it occurs than it would be for a mesophile. Psychrophiles also have polyunsaturated fatty acids in their membrane, which quench free radicals. Furthermore, psychrophiles consistently upregulate genes involved in detoxifying reactive oxygen species when compared to mesophiles. While thermophiles have enzymes with a large hydrophobic interior, psychrophiles have a small one. This keeps the amino acids in the interior moving around and preventing them from being clumped together, allowing them to continue to catalyze metabolic reactions. Psychrophiles also have more chaperone proteins than mesophiles. While hyperthermophiles use chaperone proteins to prevent other proteins from becoming denatured, psychrophiles use them to prevent proteins from misfolding and clumping together. Another threat psychrophiles have to cope with is physical damage to the cell caused by the formation of ice crystals. One simple way to cope with this is to employ compatible solutes, low-weight organic molecules such as trehalose and glycerol, in their cytoplasm, which lowers the freezing temperature of water. High concentrations of these can also scavenge the aforementioned free radicals. Apart from small molecules, Large proteins known as antifreeze proteins, or AFPs, can act as an antifreeze. They do this by binding to nascent ice crystals within the cell and inhibit them from expanding. While AFPs can't prevent water from freezing per se, the frozen water will be in the form of many small crystals versus larger ones, which can rupture the cell. Many psychrophiles also have ice-nucleating proteins, or INPs, on their outer cell membrane. These actually accelerate the rate of ice formation on the outside of the cell, and it may be the latent heat of crystallization is used to keep the internal components of the cell warm. This can be seen as a reverse sweat, where we use the latent heat of evaporation of our sweat to keep our bodies from overheating. Some psychrophiles employ extracellular polymeric substances, which are usually made of carbohydrates, but are sometimes made of proteins, lipids, phenols, and humic compounds. These can keep the water surrounding the cell liquid at freezing temperatures, again acting as an antifreeze, or limiting solute diffusion and thereby reducing freezing-induced osmotic stress. In terms of their metabolism, common metabolic pathways such as glycolysis, the TCA cycle, and the electron transport chain are downregulated, while others like the glyoxylate cycle, ethanol oxidation, acetate metabolism, and propionyl-CoA metabolism are upregulated. It may be that since the latter pathways are shorter, they will produce less reactive oxygen species, or it could be for energy conservation to enhance long-term survival. Apart from living near glaciers, psychrophiles are abundant in the expansive abyssal plains of the world's oceans, the rest of the sea bottom that's not warmed by volcanic activity. Now we'll look at microorganisms which survive in extremes of pHs, starting with acidic conditions. These are also common around volcanically powered hydrothermal systems. Magma has high concentrations of carbon dioxide and sulfur dioxide dissolved in it, and these will react with water to produce carbonic and sulfuric acid, lowering the pH. Generally speaking, an acidophile is an organism with an optimal growth pH of less than 3. The reason acidic conditions are harmful to life has to do with the way functional groups in the biomolecules interact with each other. Often, these depend on carboxyl groups having negatively charged oxygen atoms, and if they become protonated by a hydrogen ion, they will no longer interact with other functional groups in the same way. One way to limit the deleterious impacts of an acidic environment is to have a membrane that's very impermeable to protons. Apart from being less reactive, the ether-linked phospholipids characteristic of archaea also form membranes less permeable to hydrogen ions, which may explain why archaea compose a larger portion of the microbial community in acidic environments than elsewhere. Acidophilic bacteria such as Leptospirillum have a wider variety of genes involved in the synthesis of the membrane compounds than neutrophiles, and it's believed these form proton-excluding structures, but these have yet to be fully described. All organisms have to let solutes in and out of their cells, and they do this with proteins in their membranes such as porins. The ones used by acidophiles have smaller pores to limit the influx of protons, although this increased selectivity comes at the cost of being able to transport metabolites across the membrane at a slower rate. The rate of proton diffusion into the cell will be a function of the difference in pH between the cell and the outside fluid in some constant K. While the aforementioned membrane modifications can decrease K, so long as the internal pH of the cell is greater than the surrounding fluid, protons will leak in until the pH is the same inside and outside the cell. This means that if homeostasis is to be maintained, protons have to be pumped out. One way to do this is to use a reverse ATP synthase. Our ATP synthases famously use the influx of protons from a high to low concentration to power the production of ATP. Conversely, similar enzymes can use the hydrolysis of ATP to power the pumping of protons from a low concentration inside the cell to a higher concentration outside. Also, the cross-membrane pH gradient in our mitochondria is generated via the electron transport chain, where the flow of electrons from a less to more electronegative element is used to pump protons from a low to high concentration. Acidophiles can carry out these redox reactions at a greater rate to keep enough protons out of the cell, versus using them to drive their ATP synthase enzymes. When cellular biology is taught, it's commonplace to say that it's the difference in proton concentrations inside and outside the cell that drives the ATP synthase enzyme, but this is a bit of an oversimplification. It's actually driven by the proton motive force, which is a difference in the electric potential or voltage across the membrane. What generates this voltage is a difference in the concentrations of negatively and positively charged ions across the membrane. One way to decrease the proton motive force in acidic environments and the influx of protons into the cell is to pump other positively charged ions into the cell. Many acidophiles pump potassium ions into their cytoplasm to accomplish this. Within the cytoplasm itself, the production of organic acids will lower the pH of the internal environment and thereby increase the acidic stress on the organism. These acids are necessary intermediates in metabolic processes, so they can't simply be omitted, but what acidophiles often do is to produce more of the enzymes which convert these organic acids into the next intermediate, decreasing their concentration at any given time. Some of these enzymes whose upregulation is particularly prominent are the acetyl-CoA synthase and propionyl-CoA synthases, which convert the carboxyl group in acetic and propionic acid into a thioester group, along with lactate 2 monooxygenase, which converts lactate into pyruvate. The protonation of R groups in a peptide chain can lead to protein misfolding, so acidophiles will also utilize chaperone proteins to keep them in the proper configuration. Apart from volcanic hydrothermal environments, acidophiles can be found in environments where the local geochemistry leads to acidic conditions, such as acid sulfate soils and larger bodies of water like the Rio Tinto River in Spain. At the opposite end of the spectrum, organisms adapted to cope with higher pHs are known as alkalophiles. The most common aquatic basic systems are soda lakes, or small bodies of water in bedrock with high levels of sodium carbonate. These lakes are very common in the Rocky Mountains and the East African Rift Valley. Soils with high concentrations of calcite, magnesite, and dolomite will also produce a basic environment when wetted. While most hydrothermal environments are heated by interactions between water and magma, serpentinizing systems are heated when water chemically reacts with ultramafic rocks like olivine and pyroxene, which produces serpentine, magnetite, and hydrogen. While volcanic hydrothermal systems tend to be acidic, serpentinizing ones are basic with a pH of around 10. Lipid composition analysis have indicated a number of features characteristic of alkophilic microorganisms. These include a high lipid to protein ratio, along with high concentrations of cardiolipin, and a fatty acid composition consistent with a very fluid membrane. Monoacyl glycerol phosphate is present in alkaphiles and absent in neutrophiles. Furthermore, alkophilic bacilli possess squalene-based compounds with substantial quantities of C40 isoprenoids. Exactly how these features help microorganisms cope with basic conditions is yet to be understood. One thing to note is that facultative alkaphiles, which can grow in neutral conditions, have less unsaturated and branched-chain fatty acids in their membrane than obligate alkaphiles. Alkaphiles also have greater concentrations of A, B, and C-type cytochromes in their cell membranes, which may be compensating for the decreased efficiency of cellular energy metabolism. Like most organisms, alkalophiles use a proton motive force to drive their ATP synthase, yet the external concentration of protons is lower than that inside the cells. In order to generate a proton motive force, they have to expend a lot of energy to pump positively charged metal ions out of the cell. Whereas acidophiles upregulate enzymes which metabolize organic acids, alkalophiles downregulate these while upregulating enzymes which produce the acid from the prior intermediate. Some organic acids employed to counteract the external pH by alkaphiles include galacturonic, glucuronic, glutamic, and aspartic acid. Like acidophiles, alkalophiles have many chaperone proteins to prevent protein misfolding, although their main concern is ammonium groups becoming deprotonated and thereby disrupting ionic bonding. Finally, the cytoplasm of alkalophiles has a greater buffering capacity than neutrophils or acidophiles. This is largely due to a high concentration of phosphoric acid, with a second pKa of 7.2, which keeps the cytoplasm near neutral. The first of the single-directional variables we'll look at is aridity. Arid landscapes such as deserts cover roughly 41% of the Earth's land area. Life on Earth is said to be water-based, in that we use it as a universal solvent, so going without it will be very detrimental to any organism. While multicellular plants and animals frequently use strategies to limit water waste and evaporation from their tissues, Such strategies aren't very viable to most microorganisms because of their high surface area to volume ratio. Instead, the most common strategy is to adjust their life cycle to cope with the conditions present. Most microorganisms in moist soils have a continuous growth pattern, where they grow at a relatively constant rate until they are large enough to replicate. When we compare arid and moist soils, we see the metagenomes, or combined genomes of all the organisms in the environment, of the arid soils are depleted in genes involved in resource acquisition by degrading exogenous polymeric compounds or transporting smaller ones across the membrane, along with those associated with growth and competition. Meanwhile, they are enriched in genes involved in DNA replication, transcription, and translation, along with essential carbon metabolism. This suggests they're primed to have relatively low rates of activity until the environment is wetted with sporadic rainfall, at which point they will rapidly grow and divide. The metagenomes of arid soils are also enriched in genes involved in stress tolerance, like DNA repair, cation transport, osmolyte biosynthesis, and dormant spore formation. When we look at the community composition of arid soils, what is observed is that actinobacteria are more abundant compared to other environments, while cyanobacteria, proteobacteria, and plactinomyces are less abundant. Although it should be noted that many cyanobacteria form biofilms in arid environments. The cells within these biofilms secrete a gelatinous matrix to retain water, and this will result in the formation of biocrusts in dry soils. The other variety of extreme environments characterized by a scarcity of something essential for life are oligotrophic environments. Here, there is an acute shortage of nutrients primary producers need to produce biomass. Most often, these limiting nutrients are nitrogen, phosphorus, potassium on land, and iron, mostly in marine systems. While there's not much in these environments that would outright kill an organism who got blown into them, it would be very difficult for it to grow and reproduce, and in an evolutionary sense, if you can't reproduce, your survival in a given situation will be all for naught. Using spectrometers on satellites, we can determine concentrations of chlorophyll, which can be used as a proxy for productivity in marine systems. What stands out is that most productivity occurs near the coast, and the least in the middle of the oceans, or gyres. Of these, the lowest of the low is the South Pacific gyre. Cyanobacteria in these environments can use passive and active strategies to cope with the scarcity of nutrients. The passive way is to employ some principles of biochemistry. When you have a solution of substrates and enzymes and increase the concentration of substrates, the rate of the reaction will increase. Initially, this correlation will be linear, but will start to level off and asymptotically approach the Vmax value as all the active sites of the enzymes become occupied. This pattern is known as Michaelis-Menten kinetics, and apart from the Vmax, the other parameter is the half saturation constant, or Km, the concentration of substrate where the rate of the reaction is half the Vmax. The rate of the reaction will be the product of the substrate concentration and the Vmax divided by the sum of the substrate concentration and the half saturation constant. The lower the half saturation constant, the greater the affinity of the enzyme for the substrate, or the ability to metabolize the substrate at low concentrations. As a general rule, the greater the Vmax of a given concentration of enzymes, the higher the Km. This means that if an organism can metabolize substrates at low concentrations, it won't be able to rapidly metabolize them at high concentrations and vice versa. So, organisms have to face a classic evolutionary trade-off. Being able to survive in oligotrophic environments, or being able to thrive in eutrophic ones. Heterotrophic and chemoautotrophic organisms also use similarly adapted enzymes to metabolize the low concentration of dissolved carbon or other electron donors. The other passive strategy is to reduce the size of the cell, increasing the surface area over which the sparse nutrients can be absorbed relative to the size of the cell. The other active strategy is to take nutrients in other forms. For example, many microorganisms in nitrogen-limited environments are diazotrophic, meaning they have the nitrogenase enzyme, which reduces atmospheric nitrogen to ammonia. Because the triple bond in a diatomic nitrogen molecule is so strong, it takes 16 ATP molecules to produce 2 ammonia molecules. So, a large portion of the energy budget of diazotrophic organisms goes to nitrogen fixation. Since light energy is also used to drive ATP synthesis via the flow of photoexcited electrons, diazotrophic cyanobacteria generate a lot of reducing power per cell. When nitrogen is the only limiting nutrient, this reducing power is used to reduce carbon dioxide to organic carbon to be assimilated into their biomass. However, when other nutrients like phosphorus and iron are limiting, but not to the same extent as nitrogen, the rate at which carbon can be assimilated is curtailed. This is because the elemental stoichiometry of primary producers is given by the Redfield ratio, which states that for every 106 moles of carbon assimilated, there will be 16 moles of nitrogen and 1 mole of phosphorus. So, if there's a lack of phosphorus, less of the organic carbon reduced can be assimilated into the organism's biomass and thus gets excreted as dissolved organic carbon to get rid of the excess reducing power. While most of the open oceans have low concentrations of nutrients, some, such as the Bering Sea and the Southern Ocean, have high concentrations of nitrate and phosphate, yet low concentrations of chlorophyll. These are known as high nutrient, low chlorophyll, or HNLC regions. It's believed the low concentration of iron limits primary productivity in these regions. Iron plays a key role in allowing life to carry out redox chemistry, with many enzymes possessing iron factors known as ferredoxins to facilitate the flow of electrons. In iron-limiting systems, many primary producers, both cyanobacteria and eukaryotic algae, use flavins or vitamin B2 derivatives in their place. While they aren't as good as ferredoxins in facilitating redox chemistry, they're better than nothing. Now, we'll look at the unidirectional variables, where too much of something is detrimental to life. One of the most environmentally relevant of these is salinity, or high salt concentrations in aquatic environments. The ocean is of course salty, and this makes it difficult for organisms adapted to freshwater to survive in it. The concentration of sodium chloride in seawater is 0.48 moles per liter, and when you put all the salts together, there's roughly 35 grams per liter. However, there are environments far more salty than the sea. On the continents, there are salt lakes such as the Great Salt Lake in Utah. The water in here has 83.6 grams per liter of water, and even this pales in comparison to the Dead Sea, which is 33% by weight salt. Some specialized animals like the brine shrimp can live in the Great Salt Lake, but only microorganisms can survive in the Dead Sea. When bodies of water dry up, they will leave salt flats, and sometimes just beneath the salt crust, some water remains. This is the case in the Uyuni salt flat in Bolivia. Organisms which inhabit these environments are known as halophiles. Halophiles can be divided into one of three categories. Slight halophiles, which grow in water with salt concentrations of 0.2 to 0.5 molar, which includes the ocean. Moderate halophiles, which grow in concentrations between 0.5 and 2.5 molar, which includes the Great Salt Lake. And finally, extreme halophiles, which grow in water with a salt concentration between 2.5 and 5.2 moles per liter, which includes the Dead Sea. There are many halophilic bacteria spread amongst various subgroups such as cyanobacteria, proteobacteria, firmicutes, actinobacteria, spirochetes, and bacteroides, while pretty much all halophilic archaea are within a single evolutionary specialized clade known as haloarchaea, although some methanogens are also halophiles. Broadly speaking, halophiles use one of two strategies to cope with the high salinity conditions: the salt in and salt out strategies. Microorganisms who employ the salt in strategy accumulate inorganic ions intracellularly to balance the salt concentration in the environment. For slight and moderate halophiles, this involves pumping chloride ions into the cell while excluding the sodium ions, and for many extreme halophiles, potassium is pumped in as well. The uptake of chloride ions is enhanced by the presence of arginine and lysine at the end of the channel. Exactly how the potassium pumping mechanism works isn't fully described, but it does seem to involve the ATP synthase enzyme, along with a sodium proton antiporter which generates an electric potential that drives the influx of potassium. Instead of inorganic ions, many halophiles accumulate intracellular organic osmolytes such as glycerol in their cytoplasm to decrease the difference in solute concentrations between the inside and outside of the membrane. The salt-in strategy is more often employed by organisms which inhabit constantly saline environments, while the salt out strategy, where organic osmolytes are accumulated, is usually employed by organisms living in environments where the salt levels fluctuate. At the biochemical level, high salt concentrations can impact protein configurations. This is because more of the water molecules will form a lattice with the salt ions versus interacting with the amino acid R groups in the proteins. This dehydration of the proteins can cause them to precipitate out in a crystalline form, hampering their activity. One common feature of the enzymes used by halophiles is a greater abundance of glutamate and aspartate, amino acids with negatively charged R groups on their surface than non-halophiles. One possible reason for this is because it improves the ability of the enzyme to compete with the ions for water molecules, and thus make them more soluble in the solution. In order to pump ions in and out of their cells, halophiles have to expend energy. This energy can come from ATP generated via oxidizing exogenous metabolites, or it could be powered directly by light. Various proteins known as rhodopsins contain within them a pigment called retinol, which is a derivative of vitamin A. This absorbs light energy in the green wavelength range and uses it to pump ions across the membrane against the concentration gradient. The presence of bacteria with these pigments can turn saline bodies of water pink when they bloom. But while light, or radiation, is immensely beneficial to life on Earth, some varieties of it are harmful. Most famous of these is UV light, and this is an ever-present hazard to organisms living at high altitudes. Organisms which can survive with greater levels of radiation present are known as radioresistant extremophiles. Ultraviolet radiation is harmful for many reasons, and one of these is the dimerization of thymine nucleotides. One way to mitigate this employed by radioresistant microbes is to have a high GC content in their genome in a manner similar to thermophiles. However, there are other ways ultraviolet light can damage DNA, such as the formation of cyclobutane pyrimidine dimers in pyrimidine pyrimidone photoproducts, which lead to CT transition mutations. Also, as mentioned before, it can generate excess reactive oxygen species. Radioresistant extremophiles consistently show an increased expression of the genes involved in DNA repair via excision and the detoxification of reactive oxygen species. Meanwhile, they will downregulate protein expression as a whole when radiation levels increase, minimizing the damage it will cause to their molecular machinery. Also, a number of proteins exclusive to radioresistant extremophiles have been identified. For one, Deinococcus radiodurans is homologs to the Lexay regulator protein, which controls the expression of genes involved in DNA repair. Another, known as PPR, is also involved in the regulation of these genes and was induced by the presence of intense radiation. Lastly, Dictyostylum discoideum has an XPF nuclease enzyme, which can more readily identify damage in the DNA strand than other nucleases. These adaptations allow radioresistant extremophiles to survive even when irradiated with a UV light dose of 100 joules per square meter, with Deinococcus radiodurans being able to withstand 1000 joules per square meter. Apart from UV light, these adaptations have allowed these organisms to survive in artificially altered environments with high levels of gamma radiation and X-rays, along with alpha and beta particles from radioactive decay. Finally, many phototrophs inhabiting high-altitude environments employ UV-absorbing pigments. The highest of these are found around the fumaroles on Yuyako, roughly 6 kilometers high. In order for this strategy to be viable, the phototrophic algae have to grow in a biofilm with the pigments in the matrix. The last kind of extremophiles we'll look at are those who can cope with high levels of toxic chemicals in their environment. Now, this is arguably the hardest extreme variable to define, because some substances that are lethal to one kind of organism can be an essential metabolite for another. Not only that, a metabolite that is essential for an organism could be toxic for it at high enough concentrations. For example, we'll die if we go for a few minutes without oxygen, but we will suffer from oxygen toxicity if the partial pressure rises above 1.4 atmospheres. But one variety of chemicals that are harmful to virtually all organisms are heavy metals. One of the most prominent ones in the environment is lead. There are some naturally occurring environments with high levels of lead, such as certain hydrothermal systems, but human activity has resulted in lead being released into other habitats, along with other metals like copper, zinc, and cadmium. To cope with heavy metals, perhaps the most straightforward strategy is to simply pump them out. They do this using P-type ATPases, which use the energy produced via ATP hydrolysis to pump lead ions against the concentration gradient out of the cell. Apart from lead, P-type ATPases can be used to pump cadmium, zinc, and copper ions. Another method is known as intracellular bioaccumulation, which employs low molecular weight, usually between 3.5 and 14 kilodalton, cysteine-rich proteins known as metallothioneins. These bind to metal ions, and in so doing, protect the rest of the cell from their deleterious effects. Siderophores can bind to the metals either intracellularly or extracellularly, but aren't as effective at detoxifying them as metallothioneins. Other microbes sequester the metals extracellularly by secreting polymers which bind to the metals and prevent them from penetrating the cell membrane. These exopolymeric compounds are usually carbohydrates, but are sometimes proteins or humic compounds. Some bacteria adsorb the metals directly to their surface, with carboxyl groups doing this in the peptidoglycan layer of gram-positive bacteria, and the phosphate groups in the membrane of gram-negative bacteria performing the same function. Lastly, some metal-resistant bacteria are known to use bioprecipitation, where they precipitate harmful metals out of the solution as salts. For example, Providentia alcalcifaciens precipitates lead out as lead phosphate, and Klebsiella does so as lead sulfide. Another variety of chemicals harmful to most organisms are highly reactive oxidizing agents. While an oxidant, i.e., an electron acceptor, is necessary for many organisms to power their energy metabolism, various compounds like sulfite, nitrite, arsenite, and most poignant here, perchlorates, are so reactive they rapidly oxidize biomolecules. Perchlorates are produced in arid, high-altitude environments via interactions between chloride ions, water, and UV light in places like the Atacama Desert and the dry valleys of Antarctica. Apart from employing enzymes which detoxify free radicals, some microorganisms possess perchlorate reductases, which reduces perchlorates to chlorate and then to chlorite. A chlorite dismutase then produces an oxygen molecule and a chloride ion from it. In our attempts to protect ourselves from pathogenic bacteria, we have developed a number of chemicals to kill them in the environments in which we live. One strategy employed by many microorganisms exposed to these chemicals is to form a biofilm. This strategy is universally applicable because it lowers the rate at which the disinfectant can diffuse into the cytoplasm of the cells and thus give the organism time to repair any damage it causes. Another strategy is to again decrease the permeability of the membrane. When resistant strains of Pseudomonas aeruginosa are exposed to benzalkonium chloride, they will downregulate the expression of porin proteins, which allow materials in, along with lipoproteins, which keep the membrane fluid. In a manner similar to the biofilms, these modifications will limit the rate at which the disinfectant can enter the cell. As was the case for metal resistance, many bacteria will use ATP-powered efflux pumps to move the disinfectants out. The most common variety in gram-negative bacteria is RD, and in gram-positive bacteria, it's MFS. Other bacteria have developed enzymes which can break down the disinfectants themselves. For example, many microbes employ enzymes which cleave the N-alkyl bonds of quaternary ammonium compounds. The last method microorganisms employ to protect themselves against disinfectants and antibiotics is to modify the target of the agent. Many antibiotics work by binding to the ribosome and thereby disrupt protein synthesis. Mutations to the beta hairpin loop of the ribosome decrease the binding affinity of the antibiotics. Also, mutations to the ribosomal proteins will cause the antibiotic to preferentially bind to them versus the RNA, mitigating the impact it has on protein synthesis. While we may not think of areas of human habitation as extreme environments, we have nonetheless induced a situation where only specially adapted microbes can survive. So far, we've talked about each of these extreme variables individually, but in many settings, microbes have to cope with two or more of these extremes. Which extreme conditions, that of pH, temperature, aridity, etc., usually co-occur is largely determined by geologic processes. For example, volcanic hydrothermal systems are subjected to both high temperatures and acidic conditions because of the water-magma interactions. Furthermore, lower pHs increase the solubility of divalent metal ions, so microorganisms in these habitats are usually subjected to higher levels of heavy metals. Microorganisms, which can survive in environments with multiple hardships, are known as polyextremophiles. Another environment where we find polyextremophiles is on high mountains. Here, microbes are subjected to frigid temperatures and high levels of UV radiation. Deserts, apart from being arid, are often highly oligotrophic and are subjected to high levels of UV light from the lack of cloud cover. Lastly, saline bodies of water are usually basic, so a hybrid classification of extremophiles known as haloalkalophiles was outlined by microbiologists. These appear to maintain homeostasis by using sodium proton MRP-like antiporters, which pump protons in and sodium ions out. Furthermore, basic conditions cause metals to more readily precipitate out of solution, which can lead to iron limitation. But can life adapt to any combination of extremes? Recent research is indicating that it can't always do so. In the Dalal pools, which reside in the Danakil Depression of Ethiopia, the pH is 0 and the water is 50% salt by weight. When a metagenomic screening was performed on the water column, no life was detected. Now, we have found organisms in environments as salty and acidic as the Dalal pools, but no other body of water has this combination of salinity and acidity, a combination which likely renders it uninhabitable. Another environment where nothing seems to be living is the McMurdo dry valleys of Antarctica, which have high levels of perchlorates in them. Now, organisms can survive with similar levels of perchlorates, but repairing the damage they cause requires a lot of energy. It may be that in an environment as arid and oligotrophic as the dry valleys, there isn't enough energy available to repair the damage caused by the perchlorates faster than the cell incurs it, dooming any organism that lands there. So, while life may seem infinitely adaptable, there does seem to be some limit as to what environments can be inhabited that is set by very fundamental principles of biochemistry. In other words, life doesn't always find a way. Planetary bodies other than the Earth seem quite inhospitable, so to get an idea of how habitable they truly are, astrobiologists will often look at how microorganisms persist in some of the most extreme environments on Earth. However, this is often a very one-dimensional interpretation, and that astrobiologists will only look at one variable at a time. They'll argue that since microorganisms have been documented which can survive either the aridity, radiation, and oxidative stress present on Mars, life could survive there. However, given that we're finding that combinations of hardships are what make certain environments uninhabitable, while life could survive each of the aforementioned extremes individually, the combination of all of them in a single system can render it uninhabitable. If you liked what you see, please like and subscribe, and join us next time, where we apply what we discussed here to explore the habitability of Mars. Thanks.